Antigens and antibodies, structure, types, interactions in vivo and in vitro. Hello listeners and viewers. The topic of discussion here is related to basics of antigen and antibody. There are specific definitions of both the terms so as to make them understand easily. To get accustomed with the antigen, let's look upon first to concept of antigen. In immunology, an antigen or antibody generator is any substance which provokes an adaptive immune response. An antigen is often foreign or toxic to the body, for example, a bacterium which once in the body attracts and is bound to a respective and specific antibody. An antigen is a molecule that also induces an immune response in the body. Paul Ulrich coined the term antibody. The term antigen originally came from antibody generator. The antigen may originate from within the body, that is self, or from the external environment, that is non-self. The immune system is usually non-reactive against self-antigens under normal conditions and is supposed to identify and attack only non-self invaders from the outside world or modified or harmful substances present in the body under distressed conditions. An immunogen is, in analogy to the antigen, a substance that is able to provoke an immune response if injected to the body. An immunogen is able to initiate innate immune response first, later leading to the activation of the adaptive immune response, whereas an antigen is able to bind the highly variable immunoreceptor products, that is B cell receptor or T cell receptor, once these have been produced. Let us see to the concepts related to antigens. Epitope. The distinct molecular surface features of an antigen capable of being bound by an antibody antigenic molecules, normally being large biological polymers, usually present several surface features that can act as points of interaction for specific antibodies. Any such distinct molecular feature constitutes an epitope. Allergen. A substance capable of causing an allergic reaction is called an allergen. The reaction may result after exposure via ingestion, inhalation, injection or contact with the skin. Superantigen. A class of antigens that cause non-specific activation of T cells resulting in polyclonal T cell activation and massive cytokine release. Tolerogen. A substance that invokes a specific immune non-responsiveness due to its molecular form. If its molecular form is changed, a tolerogen can become an immunogen. Immunoglobulin binding protein. These proteins are capable of binding to antibodies at positions outside of the antigen binding site. That is, whereas antigens are the target of antibodies, immunoglobulin binding proteins attack the antibodies. Protein A, protein G and protein L are examples of proteins that strongly bind to various antibody isotypes. T-dependent antigen. T-dependent antigens are usually proteins. They require an assistance of T cells to induce the formation of specific antibodies. T-independent antigen. T-independent antigens are usually polysaccharides stimulating B cells directly. Immunodominant antigens are the ones that dominate in their ability to produce an immune response. It is commonly assumed that T cell responses are directed against a relatively few immunodominant epitopes, although at least in some cases, for example, infection with the malaria pathogen Plasmodium species, it is dispersed over a relatively large number of parasite antigens. Antigens can be classified in order of their class, like exogenous antigens, endogenous antigens and autoantigens. Let us discuss each of them. What are exogenous antigens? Exogenous antigens are antigens that have entered the body from the outside, for example, by inhalation, ingestion or injection. Endogenous antigens. Endogenous antigens are antigens that have been generated within previously normal cells as a result of normal cell metabolism or because of viral or intracellular bacterial infection. Let us see autoantigens. An autoantigen is usually a normal protein or complex of proteins, DNA or RNA, that is recognized by the immune system of patients suffering from a specific autoimmune disease. Now, let us have a discussion on antibody, its types and structure. Let us first understand immunoglobulin. Immunoglobulins are glycoprotein molecules that are produced by plasma cells in response to an immunogen and function as antibodies. The immunoglobulins derive their name from the finding that they migrate with globular proteins when antibody containing serum is placed in an electrical field. General functions of immunoglobulins are antigen binding and effector functions. Antigen binding. Immunoglobulins bind specifically to one or a few closely related antigens. Each immunoglobulin actually binds to a specific antigen determinant. Antigen binding by antibodies is the primary function of antibodies and can result in protection of the host. Now, let us see effector functions. Frequently, the binding of an antibody to an antigen has no direct biological effect. The significant biological effects are a consequence of secondary effector functions of antibodies. Usually, the ability to carry out a particular effector function requires that the antibody binds to its antigen. Not every immunoglobulin will mediate all effector functions. Such effector functions include fixation of complement. This results in lysis of cells and release of biologically active molecules. Binding to various cell types. Phagocytic cells, lymphocytes, platelets, mast cells and basophils have receptors that bind immunoglobulins. This binding can activate the cells to perform some functions. Now, let us concentrate on structure of immunoglobulins, that is antibodies. Antibodies are immune system related proteins called immunoglobulins. Each antibody consists of 4 polypeptides, 2 heavy chains and 2 light chains joined to form a Y-shaped molecule. The amino acid sequence in the tips of the Y varies greatly among different antibodies. These variable region composed of 110 to 130 amino acids give the antibody its specificity for binding antigen. The variable region includes the ends of the light and heavy chain. Treating the antibody with a protease can cleave this region, producing FAB or fragment antigen binding that include the variable ends of an antibody. the constant region determines the mechanism used to destroy antigen. Antibodies are divided into 5 major classes: IgM, IgG, IgA, IgD and IgE based on their constant region structure and immune function. The variable region is further subdivided into hypervariable, that is HV, and framework, that is FR regions. Hypervariable regions have a high ratio of different amino acids in a given position relative to the most common amino acid in that position. Within light and heavy chains, 3 hypervariable regions exist: HV1, HV2 and HV3. 4 FR regions which have more stable amino acid sequence separate the HV regions. The HV regions directly contact a portion of the antigen surface. The basic structure of the immunoglobulins can be studied under 5 headings, which are heavy and light chain, disulfide bonds, variable and constant regions, hinge region and domains. Heavy and light chains. All immunoglobulins have a 4-chain structure at their basic unit. They are composed of 2 identical light chains consisting of 23 kilodalton and 2 identical heavy chains ranging from 50 to 70 kilodaltons. Disulfide bonds. First, that is interchain disulfide bonds. The heavy and light chains as well as the 2 heavy chains are held together by interchain disulfide bonds and by non-covalent interactions. The number of interchain disulfide bonds varies among different immunoglobulin molecules. Second type, that is intrachain disulfide bonds. Within each of the polypeptide chains, there are also intrachain disulfide bonds. Variable and constant regions denoted by capital V and capital C. When the amino acid sequences of many different heavy chains and light chains were compared, it became clear that both the heavy and light chain could be divided into 2 regions based on variability in the amino acid sequences. These are, first, that is light chain indicated by variable light, which consists of 110 amino acids, and constant light consisting of 110 amino acids. Second, that is heavy chain consisting of variable heavy chain with 110 amino acids and constant heavy consisting from 330 to 440 amino acids. Hinge region. This is the region at which the arms of the antibody molecules forms a Y. It is called the hinge region because there is some flexibility in the molecule at this point. Domains. 3-dimensional images of the immunoglobulin molecule show that it is not straight as depicted in figure 2a. Rather, it is folded into globular regions, each of which contains an intrachain disulfide bond. Figure 2b to d. These regions are called domains. Light chain domains denoted by VL and CL, heavy chain domains denoted by VH, CH1 to CH3 or CH4. So, this was the basic structure of immunoglobulin molecule. Now, let us learn about the types of immunoglobulins. There are basically 5 types of immunoglobulin molecules known as the 5 major types of antibodies along with their properties are as follows. First, that is immunoglobulin A. Immunoglobulin A antibodies are found in areas of the body such as nose, breathing passages, digestive tract, ears, eyes and vagina. Immunoglobulin A antibodies protects the body surfaces that are exposed to outside foreign substances. These type of antibodies is also found in saliva, tears and blood. Second, that is immunoglobulin G. Immunoglobulin G antibodies are found in all body fluids. They are the smallest but most common antibody, consisting of 75% to 80% of all the antibodies in the body. Immunoglobulin G antibodies are very important in fighting bacterial and viral infections. Immunoglobulin G antibodies are the only type of antibody that can cross the placenta in a pregnant woman to help protect her baby. Third, that is immunoglobulin M. Immunoglobulin M antibodies are the largest antibody. They are found in blood and lymph fluid and are the first type of antibody made in response to any infection. Immunoglobulin M antibodies are about 5 to 10% of all the antibodies in the body. Fourth, that is immunoglobulin E. Immunoglobulin E antibodies are found in the lungs, skin and mucous membranes. They cause the body to react against foreign substances such as pollen, fungus spores and animal dander. They are involved in allergic reactions to milk, some medicines and some poisons. Fifth, that is immunoglobulin D. Immunoglobulin D antibodies are found in small amounts in the tissues that line the belly or chest. Now, let us know how antigen and antibody interacts with each other.
Action.
Antigen-antibody interactions. The first type is in vivo and the second type is in vitro antigen-antibody interaction. Let us see both of them one by one. In vivo antigen-antibody reactions. These reactions refer to the antigen-antibody reactions occurring inside the host. In the body, they form the basis of antibody-mediated immunity in infectious diseases or of tissue injury in some types of hypersensitivity and autoimmune diseases. The following are the in vivo interactions. First, complement fixation. Second, toxin neutralization. Third, virus neutralization and fourth, opsonization. Let us see what complement fixation means. Complement fixation. The term complement fixation was introduced by Ulrich. Complement was discovered as a heat-liable component of human blood plasma that increases opsonization of bacteria by antibodies and helped other antibodies to kill the bacteria. This activity was said to complement the antibacterial activity of antibody, hence the name complement has been given. It is now known that complement system is composed of a large number of serum proteins that play a major role in the animal's defensive immune response. Complement can mediate inflammation and attract and activate phagocytic cells. Generally, complement proteins amplify the effect of antibodies. The complement system is made up of at least 20 complement proteins designated as C1 to C9, C4b, C1, 1NH, S protein, properdine and others. The complement system acts in a cascade fashion, the activation of one component resulting in the activation of next. There are three pathways of complement activation: the classical pathway, the alternative pathway and the lectin pathway. The overview of complement activation provides a basis for consideration of function of complement as an integrated system during an animal's defensive effort. Next, in vivo interaction is toxin neutralization. Immunity to the disease like diphtheria depends upon the production of specific antibodies that inactivate the toxin produced by bacteria. This process is termed as total neutralization. Once neutralized, the toxin antibody complex is either unable to attack to the receptor sites on host target cells, unable to enter the cell or ingested by macrophages. An antibody capable of neutralizing a toxin or antiserum containing neutralizing antibody against a toxin is called antitoxin. Toxin-antitoxin neutralization. Neutralization of toxin by antitoxin may be demonstrated by animal inoculation. A neutralized toxin will not kill the animal, whereas a free exotoxin will either kill or harm the animal. While an appropriate mixture of toxin and antitoxin in vitro are kept for some time and injected to mice, the mixture proved to be harmless to the animals due to neutralization of toxin, and thus animal did not develop symptoms due to the effects of exotoxins. The third type of interaction is viral neutralization. Immunoglobulin G, M and A antibodies can bind to some viruses during their extracellular phase and inactivate them. This antibody mediated viral inactivation is called viral neutralization. Fixation of complement component C4b to the virus provide help in the neutralization process. Viral neutralization prevents a viral infection due to the inability of the virus to bind to its target cell. Neutralization antibodies indicate the presence of certain viruses. This test is useful in virus infection, but it can also be used to determine antibody titer to other organisms. The last in vivo interaction is opsonization. Opsonization is the process in which microorganisms or other particles are coated by antibody and or complement, thereby being prepared for recognition and ingestion by phagocytic cell. Opsonization antibodies belongs to the class of IgG and IgM and have site that to Fc receptor on the surface of macrophages. This binding forms the bridges between the phagocyte and antigen. Immunoglobulin G and M antibodies also can activate the complement system. The net result is to arrange complement C3b on phagocyte. If both antibody and C3b opsonize, binding is greatly enhanced. The second type of interaction is in vitro interaction of antigen and antibody. These reactions refer to the antigen-antibody reactions occurring outside the host. In the laboratory, they help in the diagnosis of infections, in epidemiological surveys, in the identification of infectious agents and of non-infectious agents such as enzymes. Antigen-antibody reactions in vitro are known as serological reactions. Precipitation reactions. The interaction between an antibody and a soluble antigen is aqueous solution, forms a lattice that eventually develops a visible precipitation. Antibody that forms precipitation is known as precipitin. This process is called as precipitation reaction. Formation of antigen-antibody lattice depends on the valency of both antigen and antibody. The antibody should be bivalent. The antigen must be either bivalent or polyvalent. Zone of equivalence is a point at which the maximum precipitation occurs. This reaction is widely used in several immunological techniques. Precipitation occurs in both fluid and gel. Precipitation in fluid. A quantitative precipitation reaction can be performed by placing a constant amount of antibody in a series of tubes and adding increasing amounts of antigen to the tubes. After the precipitation forms, each tube is centrifuged to pallet the precipitate, the supernatant is poured off and the amount of precipitate is measured. Plotting the amount of precipitate against increasing antigen concentrations yields a precipitin curve. Excess of either antibody or antigen interferes with maximal precipitation, which occurs in the so called equivalent zone, within which the ratio of antibody to antigen is optimal. Precipitation in gel. There are several advantages in allowing precipitation to occur in a gel rather than in a liquid medium. The reaction is visible as a distinct band of precipitation, which is stable and can be stained for preservation if necessary. As each antigen-antibody reaction gives rise to a line of precipitation, the number of different antigens in the reacting mixture can be readily observed. Immunodiffusion is usually performed in a soft 1% agar gel. Immunodiffusion divided into two types, namely simple immunodiffusion and electroimmunodiffusion. Agglutination reactions. The interaction between antibody and a particular antigen results in visible clumping called agglutination. Antibodies that produce such reactions are called agglutinins. Agglutination reactions are similar in principle to precipitation reactions. They depend on the cross linkage of polyvalent antigens. Just as an excess of antibody inhibits precipitation reactions, such excess can also inhibit agglutination reactions. This inhibition is called the prozone effect. Because prozone effects can be encountered in many types of immunoassays, understanding the basis of this phenomenon is of general importance. Several mechanisms can cause the prozone effect. First, at high antibody concentrations, the number of antibody binding sites may greatly exceed the number of epitopes. As a result, most antibodies bind antigen only univalently instead of multivalently. Antibodies that bind univalently cannot cross-link one antigen to another. Prozone effects are readily diagnosed by performing the assay at a variety of antibody or antigen concentrations. The subtypes of agglutination reactions are heme agglutination, bacterial agglutination, passive agglutination and agglutination inhibition. Now, we will discuss each of them in detail. Heme agglutination, also known as slide agglutination. Agglutination reactions are routinely performed to type red blood cells. In typing for the ABO antigens, RBCs are mixed on a slide with antisera to the A or B blood group antigens. If the antigen is present on the cells, they agglutinate, forming a visible clump on the slide. Determining of which antigens are present on donor and recipient RBCs is the basis for matching blood types for transfusions. At neutral pH, RBCs are surrounded by a negative ion cloud that makes the cells repel one another. This repulsive force is called zeta potential. Bacterial agglutination, which is also known as tube agglutination. A bacterial infection often elicits the production of serum antibodies specific for surface antigens on the bacterial cells. The presence of such antibodies can be detected by bacterial agglutination reactions. Serum from a patient thought to be infected with a given bacterial is serially diluted in an array of tubes to which the bacteria is added. The last tube showing visible agglutination will reflect the serum antibody titer of the patient. The agglutinin titer is defined as the reciprocal of the greatest serum dilution that elicits a positive agglutination reaction. For example, if serial two-fold dilutions of serum are prepared and if the dilution of 1 by 640 shows agglutination, but the dilution of 1 by 1280 does not, then the agglutination titer of the patient's serum is 640. In some cases, serum can be diluted up to 1 by 50,000 and still show agglutination of bacteria. Passive agglutination. The sensitivity and simplicity of agglutination reactions can be extended to soluble antigens by the technique of passive heme agglutination. In this technique, antigen coating red blood cells are prepared by mixing a soluble antigen with red blood cells that have been treated with tannic acid or chromium chloride, both of which promote adsorption of the antigen to the surface of the cells. Serum containing antibody is serially diluted into microtiter plate wells and the antigen coated red blood cells are then added to each well. Agglutination is assessed by the size of the characteristic spread pattern of agglutinated RBCs on the bottom of the well, like the patterns seen in agglutination reactions. Over the past several years, there have been a shift away from RBCs to synthetic particles such as latex beads as matrices for agglutination reactions. Once the antigen has been coupled to the latex beads, the preparation can either be used immediately or stored for later use. Next, that is agglutination inhibition. A modification of the agglutination reaction called agglutination inhibition provides a highly sensitive assay for small quantities of an antigen. For example, one of the early types of home pregnancy test kits included latex particles coated with human chronic gonadotropin and antibody to HCG. The addition of urine from a pregnant woman which contained HCG inhibited agglutination of the latex particles when the anti-HCG antibody was added. Thus, the absence of agglutination indicated pregnancy. Agglutination inhibition assays can also be used to determine whether an individual is using certain types of illegal drugs such as cocaine or heroin.